It's been really a lot of fun to be the seminar coordinator so far this term. And I know some of you think that I probably invited Jack to talk because of his interest in toxicity, which many of you know. I become a professor for that, and however, that's not the reason I invited. The reason that I invited Jack is that I know him a long time, a really long time, almost 30 years. And I was thinking about it this morning. The first time I met Jack, he was tiny bit small. And I was a whole lot larger. And he was in a master's program at Clemson University. I didn't have a job, but I did have an office. And we taught birds all the time. I was studying birds. And Jack admitted that he was really interested in what he was doing. I wanted to study dancing with these birds. And I remember saying something to him like, you need to know a lot more about me. I think I was reading that all those books suddenly appeared on the floor of my little office. And that was the beginning of what turned out to be absolutely remarkable career. Jack's a fellow of the American Ornithologists Union, but he's also a fellow of the prestigious Explorers Club. I've only known a few people who were really, Pat Wright is a fellow of the Explorers Club, so he's in some really good company. Who else is it? Are you an explorer? You said you weren't, which is amazing. Benita, are you an Explorers Club member? Oh, well. So, anyway, Jack is a curator and of ornithology and mammalogy at the Cal Academy. He's going to talk about the work that he is renowned for on the discovery of the first toxic bird and the work that's come after that. He's a conservationist. He's a wonderful bird watcher. He's also a mammalogist, and he's also paying attention to the Mammal University of California, which also interests me. His training is from the University of Chicago, which is where he got his PhD. And let's see, I guess you were a postdoc then at the Smithsonian. And of course, he's spent many years in New Guinea. And did you really want to start out with Birds of Paradise? You did, didn't you? It seems to me that that was the thing that you first did. And that was the first behavioral study as well. You were still at Clemson, were you? And he came back with a few marvelous photographs. And he always had, he was like this in a way. But his vision is much broader. His question, his development of his science came from something that I admire enormously, and I think a lot of us also have, and that is the serendipity of chance discovery when we're in the field that suddenly takes our imaginations and moves us forward in our science. And that's what Jack's done, and that's what he's going to talk about today, the ecology and evolution of toxicity in the guinea birds. Thank you for coming, Jack. It's wonderful to see you.
Thank you, Pat. Well, it's really great to be here. It's a
great honor, and I have to say, too, that when I was a beginning master's student at Clemson, I think the only person who actually thought that I'd be able to get to New Guinea was Patty. So I think because she believed in me, I was able to pull some of these things off. And so with that, I'll start with the story of how I first found out that the pitahou was poisonous. And I should say, too, that I am involved in a lot of different things. And one of the things that made me want to talk a little bit about this on this occasion was that Patty and I have been talking a bit about Alfred Russel Wallace, and she was involved in some talks last year, and we talked a lot about what it means to be a natural historian. And so I kind of wanted to tell the story of this research from the point of view of a natural historian and how you're motivated by what you see in the field. So rather than being theoretically oriented and theoretically motivated, you see something in the field and the animals are telling you that something here is interesting and something needs to be paid attention to. And then how one formulates the questions around that and chases them down one by one. And so this all started when I was a graduate student. And I was at Clemson University studying birds of paradise, and we had about 40 nets up in the forest. And as we were walking through the forest, letting loose all the birds we didn't want to catch, this was one of them. It's a hooded pitahui. It's brightly colored. It's a J-sized bird with very sharp beak and claws, and they can cut your hands up when you're taking them out of the nets. And so the first thing that you do when you have 40 nets to run is you lick your fingers and you run to the next net. And after doing this, after handling the hooded pitahui, your mouth begins to tingle and burn. And I like to explain the sensation is a lot like putting a 9-volt battery on your tongue. It's a pretty unpleasant feeling. And I realized, well, you know, something's going on here. And I thought maybe I had brushed up against some poisonous plant, or I had too much DEET on my hands, or whatever. And then when it happened again to one of my field assistants, we put our stories together, and we realized the only thing that we had in common was that we had both been taking pitahuis out of the net. We thought, wouldn't it be bizarre if these birds were poisonous? So the next time we caught one, we simply plucked a feather, popped it on our tongue, and tasted it, and there was that same feeling again. So the next thing we did was we talked to our local field guides, and we asked them what they knew about it, and they said, whoa, you shouldn't be touching those birds. Those are rubbish birds. You should leave those alone and focus on birds of paradise, which are much more beautiful and interesting. And we said, no, actually, we want to go study these birds. So we got some permission to study them, and what I'm going to be talking about today is what we've learned about some of these topics here. So first of all, what toxins the pitahuis carry and how we figured that one out, and what are the toxins' pharmacological effects, and that's important to understanding how they might be able to use it. Then also, how are pitahui toxins distributed? So both within the body of a single individual, and what questions can we answer by knowing that? And then also among the different species of pitahui and among their geographical range, and what can that tell us about the natural history of these toxins? Then you want to begin talking about, well, are they actually defensive? Can they be used for chemical defense, and how we investigated some of that. Then what's the source of pitahui toxins? Where do they come from? And then a little bit of work that I've done recently on the phylogeny of pitahuis and what that's told us about the evolution of toxins. And finally, something that we're really just sinking our teeth into now, and I'll just tell you where we're going with it, and that is, how do pitahuis tolerate such high toxin levels in their own tissues? So as an introduction, this is the genus Pitahui. There are 6 different species: the hooded pitahui, the variable pitahui, white-bellied, rusty, black, and crested pitahui when we first started our work. And these are all omnivorous, J-sized birds, and they're all endemic to the island of New Guinea, which is on the western end of the Pacific Ocean, the eastern end of the Indonesian archipelago, and just north of Australia. And so that's the only place to find these birds. So first of all, we wanted to ask, well, what chemicals are involved, and what toxins are there in the pitahui's skin, and then what's the toxin's pharmacology? How do they act? How are they toxic? So in order to do this, once we realized that these birds were poisonous, we went down to the capital and we got permission to collect a handful. And we collected 2 to 3 pitahuis from 3 different species: the hooded, the variable, and the rusty pitahuis. And then I spent a devil of a time, about almost 2 years, trying to find a chemist who believed in me and these birds that they might be poisonous enough to actually have a look at them. And we got very lucky to work with John Daly and Tom Spondy and Martin Garofo of NIH. And they're the ones who've been working on frog poisons for a very long time. And that was serendipitous for a number of different reasons. But then what we did was we did a number of different fractionations. So we would try and purify the chemical, and each time trying to figure out where the toxicity migrated with which fraction. And we found that an alkaloid fractionation was quite efficient at purifying the toxins, and that they behaved as alkaloids. Then we tried to shoot them on a gas chromatograph mass spec, a GC mass spec, and we found nothing coming out the other end, and that suggested that it was a non-volatile compound. So we dried it on a direct probe, and we used electron impact mass spectroscopy to try and figure out what it was. And the spectrum that we got at the other end, John Daly immediately recognized and said, I think that's batrachotoxin. And so in order to cinch the identification of batrachotoxin, we used a simple thin layer chromatography, where our 2 unknowns were in lane 3 and 4, and batrachotoxin standards were in lane 1 and 2, and then you simply put this end of it into some methanol and chloroform, and then your chemicals migrate this direction, and our unknowns stain the same blue reaction with modified Ehrlich's reagent as batrachotoxin and homobatrachotoxin standards, and this shows that we have quite a bit of this chemical and that it matches homobatrachotoxin. Now, as the name implies, batrachotoxins come from frogs, batracho meaning frog. So these were toxins that were well known, and these are steroidal alkaloid neurotoxins. So let's walk through what that means. So the base of the chemical, this part here with the 4 rings, is a steroid, and so it's related to things like cholesterol and testosterone. It's an alkaloid because of this nitrogen-containing ring that also has the oxygen in it, and that's a basic nitrogen, so that's what allows it to migrate into the alkaloid fraction when we do the acid-base fractionation. And it's a neurotoxin. It's neurotoxic because it binds to the sodium channels in nerve and muscle membranes, and these are the channels that open and close in a pulse down the nerve ending, or the nerve axon, and that actually sends the electric signal down. And the electric signal is actually sent by the opening and closing of these channels. Batrachotoxin binds to the channel and stabilizes it in the open conformation. So once the sodium channel is activated, it remains open, and it allows sodium to flow freely across the membrane. And this prevents the cell from reestablishing the ionic potential that's needed for the next firing. So it basically incapacitates the nerve and muscle membranes. It's among the most toxic natural substances known because most other toxins that bind to sodium channels actually close the channel, and so the other channels around it can continue to send that pulse until enough of them are actually incapacitated. But you don't have to open that many of the channels to depolarize the cell. So it's much more toxic than strychnine, curare, and many of the other things that you think of as equal to poisoning darts. And it's also expected to affect a wide range of species because the batrachotoxin binding site is highly conserved on the sodium channel. So basically everything that has a central nervous system that's been tested has been found to be sensitive to batrachotoxin, with only a couple of exceptions. So as the name implies, these toxins were previously known from frogs, and only of the genus Phyllobates. This is Phyllobates lugubris, Vittatus and Aratania, Phyllobates bicolor here, which has some geographic variation, and then Phyllobates terribilis. And of all the so-called poison dart frogs, only 3 species were ever actually used for poisoning dart tips, and all of them are in the genus Phyllobates. They're Phyllobates Aratania, bicolor, and terribilis, and they all have exactly the same toxins that the poisoners carry. Not similar, but the same toxins. A couple of things that are interesting about these frogs, and that we want to begin thinking about with the birds, some of the questions that they raise. Well, first of all, these frogs are found only in Central and South America, and the toxic ones, the most toxic ones, are restricted to the western Colombian lowlands. And so, there's no exchange of toxins between pitahuis and these frogs. From what we know about the frogs, they do not produce the toxins themselves. When you bring them into captivity, they slowly lose their toxicity with time. If you raise them in captivity, they don't produce the toxins at all, but if you feed them the toxins, they can readily accumulate it in their skin. The frogs have specialized skin glands that are most concentrated on their backs, just behind the head, and that allows them to exude the toxins when they want to. And so that's another thing we want to begin thinking about with the birds. We want to think, do they have any specialized glands or ways to store these? And the frogs appear to be totally insensitive to the toxins, and that's another question that we have of the pitahuis is, are they also insensitive to the toxins, and how do they do that? So then, now that we knew what the toxin was, and because we knew what the toxin was, we had a very sensitive radioligand binding assay that had been developed in the past that we were able to use. And so we could ask this next series of questions, which is, how are the toxins distributed within a single individual bird? And what can that actually tell us? Well, one, we might be interested in which part of the bird actually stores the toxin, and are they being stored away from the tissues that are normally poisoned, and might they be available for chemical defense? So these are some of the things that we might be able to ask by doing a simple survey of the toxicity across the body of a bird. So to do this, we got permission to collect 20 individuals of hooded pitahuis, and in the field we separated each one of the bodies into all these different tissue types. So feathers were put in little Ziploc baggies, skin, internal organs, striated muscle, and saliva and preen glands were all separated and put in their own vials with ethanol, and then brought back to the laboratory. Now, saliva and preen glands were interesting to us because we knew that the toxin was in the feathers, and we were thinking of what are the different parts of the bird body that produces secretions that are used in preening. And saliva is definitely one, and the preen gland, also known as the uripedial gland that's on the rump of the bird, produces a number of different oils that the bird uses for preening. So just to walk you through this first graph, and there'll be a few like this, but the y-axis here is on the scale of microgram of batrachotoxin equivalents per gram of tissue. So this is a measure of toxicity. It's not actually a direct measure of how much toxin there is, but how much toxicity there is in the radioligand binding assay per gram of tissue. And then here on the x-axis, we have the different tissue types that we screened: skin, feathers, heart, liver, muscle. Uropygial gland and saliva glands had no detectable amount of the toxin, so they weren't even included here. And then up in the top, these are a posteriori Bonferroni corrected paired t-tests to figure out which of these different tissue types were significantly different from the other in toxicity. So first of all, the uropygial and saliva glands had virtually no toxin in them. And so that was not the source of the toxin and not a storage organ. Next, let's look at muscle and heart and liver. These are important because they have the requisite sodium channels, and they should be poisoned by the toxins if the birds are sensitive to the toxin. So muscle, even though it had the least amount of toxin, just to put this into perspective, 0.05 micrograms of batrachotoxin, if injected into the hindquarters of a mouse, will kill it within a minute. So every gram of muscle has about enough toxin to kill 100 mice. So even though this doesn't look very impressive and it's the smallest bar on the chart, it definitely suggests that pituhuis are insensitive to the toxin somehow. And then the next thing I want you to look at is the skin and feathers here. And skin and feathers are both on the outside of the bird, and that's where the toxins are most concentrated. So if we want to entertain the possibility that these birds might be using it for chemical defense, the most likely or useful places to store the chemicals or have them available would be on the skin and feathers on the outside of the bird. And so we did want to begin exploring the possibility that birds might be using this for chemical defense. Now also, because skin was the most toxic tissue, we were interested in looking at skin a little bit more closely. And so we did some work early on, and we were able to get some samples from the field, from some of those early collections that were fixed in the field in modified Karnofsky reagent, and then brought back and formalin fixed and sliced for a transmission electron micrograph work. And what we found is that pituhui skin is a very typical bird skin, and it looks just like other birds. There's no special sweat glands or oil glands in bird skin, and I'll talk a little bit more about how birds move lipids to the outside of the skin. But it turned out that of all the samples that we had collected, this one that we had fixed was actually a relatively less toxic sample. So later we were able to get another more toxic sample, and we just analyzed this a couple years ago. And when we looked at this one in the lab, it was really striking. So in fact, I should walk you through this picture first. So that would be the outside of the bird here. This is the thickened stratum corneum. This is the cornified layer on the outside of the skin. And then this is the epidermis here and the dermis. Then there are a lot of different things inside the cell, but you'll get a better look at them here. So when we had a more toxic individual, and again, that would be the outside of the bird, the stratum corneum, and then the dermis here, we saw these really dark inclusions that were very puzzling, and my colleagues who've been studying bird skin for many, many years had never seen anything like this, so they were really intrigued by it. When they looked more closely, it turns out that these things are uncharacteristically electron dense, and they're multigranular bodies, which are analogous to lamellar bodies in mammals. And if we look at them even more, you'll see that there's this lamellar structure. And in birds, let's see, there's one more here. In birds, what happens is that these multigranular bodies will begin to coalesce as they move towards the surface of the skin. They lose the lamellar structure, and they form these lipid droplets that are exuded into the layers of the stratum corneum. And this is how birds retain their waterproof ability, if you will. So that's how they keep water from coming through their cell, through their skin. Well, it turns out that the stain that we used is osmium tetroxide, and it's a very standard stain that's used for this kind of work. But it binds very strongly to the pyrrole ring in batrachotoxin and homobatrachotoxin. And so it turned out to be a very good stain for batrachotoxin, and it was actually telling us that the toxins are mostly in these multigranular bodies. And there's one other here. This is interesting because at first we thought that maybe the batrachotoxins were just passively sequestered into lipid-rich tissues because they're also lipophilic. But it turns out that many of the other lipid-rich elements, like mitochondria, which has a lot of membranes in it, it doesn't appear to be going to those organelles at all, and it seems to be localized in these multigranular bodies. and then that has inbuilt mechanisms for moving the toxin into the stratum corneum on the surface of the skin. So basically, the batrachotoxin binds with osmium tetroxide, and we tend to find the batrachotoxin in the multigranular bodies in the skin. So this is something that we're still following up on, and we're going to try and get more samples in the future to do more work on this. But the summary of the work that we did on these different tissue types is that the muscle appears to be insensitive to batrachotoxin. And after we realized that, John Daly sent me to the field with a little sample of about 50 micrograms of batrachotoxin, and we injected that into the leg of a pitahui, and that would be enough to kill you or me. And the pitahui showed absolutely no effect to that at all. So we're quite convinced that the pitahuis are totally insensitive to the batrachotoxin. Batrachotoxin is most concentrated in the skin and feathers on the outside of the bird, where it may be useful for defense. And multigranular bodies of avian skin seem to be involved in toxin accumulation and secretion. So then the next questions that we wanted to ask involved looking at toxins distributed across the different species of pitahuis and also across the geographical range of pitahuis. And there's a couple different reasons that we're interested in this. So first we might ask, well, are all pitahui species toxic? So some of them are more brightly colored than others. We are interested in whether or not this bright coloration might be correlated with toxicity. We're also just interested in when toxicity might have evolved in the genus. Among localities, we were very interested in this, especially after talking to folks like Jared Diamond, who have traveled to many different parts of New Guinea. And you know, some of the colleagues that we have were a little bit skeptical. They said, you know, I've traveled a lot in New Guinea, and we've never been told by the local people that these birds are poisonous. I've never experienced this myself. Could it just be a local epiphenomenon, or is this something that's more widespread? So we were very interested in whether or not this might be true. And one thing that we know about other species that seem to be geographically variable in their toxicity level is that they may not have equal accessibility to the toxin in different regions. So for example, monarch butterflies, when they're reared on toxic milkweed plants, they become very toxic, but if they happen to be reared on non-toxic plants, they tend to be non-toxic. And we are hoping that this might help us. If there is variability, it might help us find the source of the toxin that pitahuis are using. So to do this work, we were able to collect samples from all 6 of the pitahui species that were known at the time. And in order to sample in some places where we weren't able to collect whole birds, we used feather toxicity as a proxy for overall toxicity, and we used the same radioligand binding assay to do a lot of this work. So here on this graph, these are the 16 different villages or localities in New Guinea that we sampled. These are 5 of the 6 pitahui species here. And then again, this is our measure of tissue toxin concentration on the y-axis. So if we sort of look down the graph in this direction first, this is what we see. So what we found was that there appears to be 3 different toxin classes in the genus Pitahui. The white-bellied Pitahui appeared to have no measurable amount of toxin. We couldn't bind this away from 0, so they appear to be more or less non-toxic.
Jack, they're all monomorphic, sexually monomorphic?
No. These all are, and the hooded pitahui is, but the variable pitahui, because its name is variable pitahui, there's over 20 different subspecies that have been named, and they vary tremendously. Some of them look more like the rusty pitahui, some of them are almost perfect mimics of the hooded pitahui, and 2 or 3 of those races are sexually dichromatic. So you see brightly colored males and dull females. And then you see others where both the males and females are brightly colored. So, yeah. Yeah, no, yes, it is an average, and yes, there's a lot of variation. In fact, I should say here that each one of these bars is where we were able to collect a species. Many of these localities only have 1 or 2 of the species, so wherever there's a blank space, it's telling us that that species didn't occur in that locality, or we weren't able to collect it there. There would be a bar every place that we had sampled them, with the exception of the white-bellied pitahui that was never toxic, and we only sampled it in one place. We didn't want to use up the space. So yes, there's a tremendous amount of variability between the different localities and even between the tissues. And so we published a paper on this, and there's excruciating detail in my thesis that has a lot of this in here. So we did so many different analyses of variants to try and understand this, but there's even an interaction effect between the 2 tissues. And maybe I'll mention a little bit more about that in a minute. So let's see. And then this is a less toxic class, and then these would be the most toxic birds. And what we do see here is, superficially at least, these are the most brightly colored, these are less brightly colored. But we'll talk about that more too later in the talk. And just to put this into perspective, these birds in most of the localities, but not all, but most of the localities, you could pluck a single feather, pop it on your tongue, and get that tingling, burning sensation. That was sort of our rough field bioassay that we used all the time. And there was one place that the birds were so toxic that just holding them in your hand and taking them out of the nets would cause your eyes to water and your nose to run. And the local people in that village said that they were allergic to them. And the sensation that you get is very, very similar. And you get puffy eyes, and that sensation lasts up to a day. That was never experienced with any of these birds. And we were never able to taste the toxicity with a single feather, although sometimes if you actually tasted the skin, you could get a little bit of tingling and burning sensation. Okay, so to come back to this slide, and now let's look across localities. So one thing that we found was in every locality that we sampled birds, there was some level of toxicity. So they were toxic everywhere, but there was a great deal of variation. Now, another thing that's interesting about this, Brad, I think this might come back to your question, is that we only had a chance to visit each one of these villages once. And so one thing that's also confounded in this is that if they're moving it from their skin to their feathers, and we don't know much about feather development or how it gets put into the feathers, there could be effects of that too, effects of time of year. So this is really a very broad brush and just the first chance that we had to look at it. But what we did find was that there's toxicity everywhere, but there's tremendous variation, which might be useful in figuring out where the toxins come from. I actually had a student at UC Berkeley for a couple years who had set up a really clever project to raise birds in Goroka in the highlands, and he built a little aviary, and his plan was to watch the feathers grow, and he could pluck feathers, and that would prompt new feathers to grow. And then we were interested in whether feathers grew toxic and then the toxins wore off with time, or whether they grew non-toxic and they accumulated with time from preening, and what this might mean for the birds. But New Guinea being what it is, it was very hard to do that work, and his project, he ended up moving to another project after that. But if any graduate students are looking for ideas, we can talk later. So all that variation was, again, another sort of indirect suggestion that the birds might be getting their toxin from some environmental source. And now as we were traveling from village to village, and we would go and find the best guides and the big man of the village, and we would ask them, so we've come to study poisonous birds, would that be okay? And they'd say, well, yes, but which poisonous birds do you want to study? And we'd say, well, which ones have you got? And this was a bird that many of the villages that were near this habitat, they said this bird was poisonous too. This is called the blue-capped Ephrata, and we were able to collect a handful of these too. And when we brought them back, not only did they contain batrachotoxin, but we were able to describe 3 new chemicals, chemical forms of batrachotoxin: the cis-crotonate, the hydroxypentanoate, and the acetate. And interestingly, once we knew what these were and we had the tools for identifying them, we went back to the earlier bird samples and found them, and to some of the poison dart frog samples and found them there too. So the fact that they were found in this distantly related Ephrata bird and the pitahuis suggested to me, at least at the time, that it was most likely that they were going to be getting their toxins from some environmental source, rather than independently evolving the machinery to produce these very complex chemicals. So the next question that we wanted to try and address was, can toxins protect pitahuis from their enemies? So can it be useful for defense? So the first thing you have to begin thinking about is, well, what enemy? Which enemy are we talking about? And birds have many different enemies. So one of the first ones we began to look at was ectoparasites, and they would be an important class of enemies for birds because they live and feed and breed on skin and feathers, which are the most toxic tissues of the birds. They would be expected to have voltage-gated sodium channels. It would be expected to be sensitive to batrachotoxin. And we know that ectoparasites have significant fitness effect on birds. They can affect egg numbers and hatching rates. They can affect mating success and mate choice decisions. They can vector important avian diseases, and they can just contribute to the overall discomfort of birds. And so we think that these are relevant to be studying. And this is actually an ear infection that's not unusual And so these are actually small ear mites that we would often find on pitahuis, and this is actually a hooded pitahui's ear right there. So in order to do this work, we collected Theraptera lice from birds in the habitat, and we used them because we could catch sufficient numbers of them to do the work that we wanted to do some nice experiments. They were relatively easy to keep in the laboratory and large enough for us to easily see and score. All the lice that we used in these experiments were Theraptera lice from two main classes, either Amblyceran and Ischnoceran lice, and we were able to classify them better to their genera. And we were able to block, because these are very different types of parasites, we were able to block the effects. So the first type of effect I like to think of that would be helpful if you're going to be chemically defended, I like to think of as an insect repellent type effect. So if you could repel these ectoparasites or try and keep them off of you, deter them, that'd be super handy. And so what we did was we simply collected a lot of these lice and we put them into petri dishes and we gave them a choice of two different feathers, one toxic and one non-toxic. And every hour we went back to the petri dish and we scored which feather they were spending their time on. And we continued to follow them until the lice died. And we had a total of 105 lice that participated in these first experiments. And we compared the hooded pitahui, the highly toxic feathers of hooded pitahui, to non-toxic control feathers. And these showed a very strong aversion to the pitahui feathers. So most of these lice spent their time on the non-toxic feather. And then we were interested in whether or not the toxin levels of the less toxic birds would be helpful as an insect repellent as well. So we compared feathers from the rusty pitahui and crested pitahui, which are both low toxicity, to the little shrike thrush. And here we found no significant effect. So toxicity at that level did not seem to affect these Theraptera lice. But if you think about it, most lice, they hatch on a single host and they don't have opportunities to go exploring other hosts and making these kinds of decisions or choices. And so we wanted to think about, another type of effect, which I like to think of as an insecticide effect. So they might actually kill the lice, or even if they're not actually killing the lice, if they can just shorten the lifespan, that significantly reduces the impact of each louse. So we basically repeated those experiments in the petri dish, but this time gave the louse only one choice, and it would either be a toxic or non-toxic feather. And then we measured the lifespan, how long those lice lived in the petri dish. So here's our non-pitahui, rusty pitahui, and hooded pitahui, and the average louse lifespan, and some bars here. And I can't even include the error bars here because there are so many different types of lice that the error bars are kind of meaningless. But we were able to do all the blocking that was necessary to look for a statistically significant effect. And what we did find was that the lice live a significantly longer period of time on the non-pitahuis than they do on the rusted pitahuis or the hooded pitahuis. And so in this case, it does seem like there's a significant insecticide effect of the toxins, which might be useful. So the bright coloration of birds, if it's useful for chemical defense at all, wouldn't be to signal lice, though. But one of the things that's so interesting about batrachotoxin is it would be such a broad spectrum toxin that we should be thinking about many different potential targets of batrachotoxin. So in New Guinea, there are several different snakes that feed on birds the size of pitahuis. These include green tree python, amethystine python, and Daubertese python, and then of course the brown tree snake, Boiga regularis. So this was, many of you will be familiar with this snake. This is the one that was accidentally introduced to Guam during World War II and has decimated the vertebrate populations there. And this snake is endemic to the island of New Guinea, something that these birds would have had to deal with for millions of years probably. And so we wanted to find out how these snakes would respond to batrachotoxin. So we did a couple very simple experiments, and these were very short-lived. But what we did was we basically took their normal prey, which was either pinky mice for the captive Boiga and day-old chicks for the green tree pythons, and we sprayed them with a small amount of batrachotoxin that would be about a quarter of what you would expect in pitahui's feathers. And in every case, what we found is that the snake actually bit the prey item and then wrapped itself around it and began to ingest it. But it usually would just get a short distance and then it would wrap itself back around the prey and try and pull it out, and which can be really difficult for the pythons especially. But they were able to get it out, and then they would leave the prey back in the corner of the cage, and then they would be readjusting their jaws. Some of them became so visibly infused with mucus that they were literally blowing bubbles through their own mucus. And they wouldn't eat the prey. But in every case, they did kill the prey. And so it might not be very helpful to that particular individual, but perhaps others. So what we think from all this is that it's very possible that if the young in the nest have any botryga toxin on the outside of them, that if a snake would walk up a branch and tongue flick or even kill one young, if there was another one in the nest, it might be spared.
Jack,
But
did you ever do an experiment of showing a snake that had suffered from this another?
we did.
Did they go after it again?
If time had passed and it was like the next day, and usually it'd be a week before we would do it again. And the Boiga were the only ones that we actually did multiple times, and we didn't see any sign of learning.
Oh, so they're dumb.
So, but again, and here's another thing about trying to work with birds like this in New Guinea is I'm always envious of my colleagues that have shoe boxes full of poison dart frogs in the laboratory, or who study plants in the greenhouse and can have hundreds and hundreds of replicates. It's very hard to do these kinds of things. So we're kind of limited to some rough inference, and that's really all I can tell you about the snakes. Is that we do know that they're sensitive, that they do respond very strongly to the toxins, but there needs to be better experiments of how the snakes actually respond and learn if they actually do.
Jack,
Yeah.
before you leave this, do you happen to know where the Boiga, I mean, you probably know the regular is in Australia, it's been introduced to Guam.
Yeah, the experiments that we had done were at the National Zoo at Smithsonian, and they were all from Guam.
So they were field collected from Guam.
They were field collected, but they were from Guam, which were probably ultimately either from Manus Island or mainland New Guinea. I don't think anyone is 100% sure about that. You might be able to tell me. Yeah. So it might not even been the most appropriate test, but it was what we could get our hands on. So likewise, we have even less data about hawks, but I just want to mention them because there are at least 6 different species, actually 8 different species of hawk in New Guinea that are large enough to take a pitahui-sized bird. And they should have the requisite sodium channels, and they should be poisoned by the toxins, and they're probably better learners. But we've done no experiments, and it's very hard to witness predation events in the field with hawks. And we had explored the possibility of putting up some mounted birds and painting them, and in reality, it would be very hard to publish anything if we had painted them ourselves. And our ability to get and kill pitahuis just for these experiments we really couldn't do it, and we didn't want to do it. So we don't have any direct evidence, but we do think that these would be likely targets of the toxin as well. What we do have a lot of data on is human hunters. And in most of the villages I've been to and stayed in, the human hunters are amazing with their bow and arrow. I've seen them pick a bat out of the air with a bow and arrow. And most of the birds are hunted out from around the village and in the local gardens by the local hunters. In every village I've been to, the local people know not to eat the pitahuis. In most places they say, yeah, it's because they're poisonous. But I have been to 2 different villages where they just tell me it's a taboo and they know not to eat them, but if I ask them, why don't you eat them, they just say it's taboo. We don't know. It's just taboo. So it may have been something that's so ensconced in their livelihood that they know not to do it, but they've forgotten why. And there are actually several different ethnographic accounts that were published, and a couple of them before we published our first paper, saying that the pitahuis were poisonous and the local people claiming that they were poisonous. So we know that the local people know about it. We know that the local people avoid them and don't hunt them, and that they would be protected from the local people because of the toxins. We don't know how much of an evolutionary impact that would be. The next question that we wanted to attack was, where do the toxins come from? And this is a really significant problem when you're dealing with a large omnivorous bird in one of the most diverse forests in the world, trying to figure out what it is they're eating that might have the toxin in it. And we spent several years looking for this, but we could never just focus solely on that. But we had to design other projects that would get us out to the field, that would allow us to do a lot of collecting. And I ended up spending about several months in this little tiny village called Harawana that's a long plane flight away from even the nearest road end, and worked with these folks in 1995. And after being held at gunpoint by bandits so many times that we just gave up our other field site, I sent my students out to Harawana. And when they got there, the local folks were like, we knew you were going to come back, Jack, and we're so excited that you're here because while you were away, we figured out where the Pitahuis get their poison from. And they had this amazing collection of about 40 vials, and every vial had multiple little beetles in them, and the name of the collector, the name of the locality, and the date. And this was never something that I asked them to do, but they watched what we scientists do in the field, and they mimicked it very nicely. And
Well, here's the case where the cargo cult science thing actually worked.
And the person who actually ran that study, who speaks no English at all, is the second author on the PNAS paper where we published this. So that was one of the funnest papers that I've gotten to write about Pitahuis in a long time. So the story is that in that local village, they call the blue captifrida the nanny sanny bird. And when I was there in '95, they said, yeah, nanny sanny means bitter, so it's the bitter bird. And they even had a name for that. Well, it turns out that nanny sanny refers to the specific, they have other names for bitter as well, and nanny sanny refers to the specific sensation that you get when you taste this. And the older gentleman in the village knew that there was a nanny sanny beetle too, and if you tasted that beetle, you got exactly the same reaction. And in fact, they said that you could often find the beetle when you were cutting grass for thatched roofs, and if you're really sweating and it gets on your head and the sweat runs into your eyes, they said, that's bad. That's no good. And so this is what the beetle looks like, and it's about the size of a grain of rice. It's tiny, and it would mean almost nothing to the birds in terms of calories. So it would be an insignificant amount of their diet. And they're very hard to catch because they don't seem to congregate. In fact, when we've looked at stomach contents of these, they appear to be eating even smaller things. So they're probably predatory, and they spread out. It's in the genus Coracine in the family Maliridae. And when you look at these a little bit more closely, you see that they have these interesting glandular vesicles on the sides of their bodies, and that when you hassle them, they curl up and they exude this little liquid substance out of these vesicles. And although this has been reported for the family in the literature, almost nothing is known about what they do with those glandular secretions or what's in those glandular secretions. As far as I know, it's never been studied. But there is one account, Patty, that you would enjoy, that apparently the males will do this and feed them to the females when they're courting them. So if they are actually toxins, that would be a useful thing to give to the female to help defend her. Then we had to go back and figure out whether the Pitahuis are actually eating these. And we had collected dozens of stomachs and stomach contents from Pitahuis from the earlier part of our work. So we basically were able to show that the Coracine beetles contain significant amounts of the Batrachia toxin. When we looked back through these birds' stomachs, they contained up to 30% insect matter, often containing Coracine-sized insects. So we knew that they at least took Coracine-sized insects. And we found one stomach that had a Coracine beetle in it. It was from a different species, from a different locality. But we did find that they do eat the Coracine beetles. And the indigenous naturalists provided the key insights to do that. So we show that the beetles have the Batrachia toxin, the birds eat the beetles, and we think that that's likely a source, that although it's still a possibility that both the birds and beetles are getting them from some other source.
If the beetle males are giving it to the beetle females, that suggests that the beetle females can't get it, or they don't produce it, or they don't produce enough of it, or what?
Well,
Is it also an environmental source for the beetles?
we think it may be. There's actually a paper published a few years ago suggesting that many of the poison dart frog compounds actually may come from orobatid mites. And that when you screen orobatid soil mites, you get more of the different poison dart frog compounds than if you screen ants. And that maybe even the ants are getting them from orobatid mites. And it was a very intriguing paper, and they had some great chemistry in there. But there's a lot of work that needs to be done on the sources of those, of the poison dart frog poisons and these. And they've never figured out where Batrachia toxin comes from for the poison for the Phyllobates frogs. So even though they found some of the pumilio toxins and the quinolizidines and pyrrolizidine alkaloids in ants, they've never found Batrachia toxin in ants. And until the Pitahuis were found, they'd never found it anywhere besides the poison dart frogs. And now we have them in Pitahuis, Aphridas, and these Coracine beetles. And as I mentioned, the Coracine beetles, which are about the size of a grain of rice, they appear to be predatory. So if they're getting it from something else, it's probably something even smaller, like orobatid mites or thrips or who knows what. It is possible that they're making it themselves, but apparently beetles don't have the mavonolate pathway that's needed to make steroids. So if they're making it, they would have to get the steroids from someplace else. So.
Or about as mites too are all brightly colored, most of them are.
Yep. And, and apparently they're a big dietary source for the smaller poison dart frogs. So let's see, I got to move along. So then this prompted us to do a little bit of phylogenetic work to understand the evolution of the genus Pitahui and maybe map on their bright coloration and map on there the evolution of toxicity. So again, here are the 6 different species of Pitahuis that we considered. Just a reminder that the hooded and variable Pitahuis were more toxic than the others. And when we first did our phylogeny, we realized that the genus Pitahui is polyphyletic. Then we were able to get more samples from more different things, and I realized that this isn't going to be easy to see. But basically, the crested Pitahui turns out to be sister to the bellbird of Australia. The hooded and variable Pitahuis form their own group, which we couldn't link to anything else that we knew of. The black Pitahui turns out to be sister to the whistlers, the Pachycephalidae, the Pachycephala in particular. And the white-throated and rusty Pitahuis turn out to be sister to the little shrike thrush, Collioricincla megarinca. And the Ephrida turns out to be fairly closely related to Melampida in the birds of paradise. So these are very distantly related groups, and our taxonomic sampling was relatively small, but even with our biased taxonomic sampling, when we took the individuals that we had tested for toxicity, so the toxic ones that here are all in black, and then the others that we tested for toxicity like Ragologus, Eulocystoma, Aleidrias, and a couple of the others, we were able to do a very simple mapping of ancestral character states, and we used Bayes traits to try and assign some of the key ancestors. And what the analyses suggest is that toxicity is not ancestral, that it probably evolved multiple times on the tree. And when you actually take all the diversity of passerine, so this is basically doing a consensus of our tree and Keith Barker's 2004 tree, you can see even better how spread out the Pitahuis are and how distantly related they are. I've talked to Jared Diamond a little bit about this, and the fact that they've been put in the same genus for so long might actually be a case of Müllerian mimicry in birds. So they've all evolved to look so similar and act so similar in the field because they're all toxic, and it would behoove them to all work together. And we have a little paper on Müllerian mimicry that I don't have time to talk about today, but you can find it at my website. So you can see here the rusty Pitahui on top with Collioricincla, which is the little shrike thrush. Pitahui nigrescens is the black Pitahui with the whistlers. Ephrida is very distantly related. And here's the hooded and variable Pitahui way out on a limb. And it took another team of Johnson et al., and this was just published in 2010, to try and figure out what Pitahui's real sister group is. And it turns out that they're related to the Old World orioles, and they're probably the sister taxa to all the Old World orioles. And so this really throws a wrench into thinking about the bright color. Well, maybe it does, maybe it doesn't. You have to, but it turns out that there's some orioles that look just like that, which is remarkably similar to what a hooded Pitahui looks like. And some of these are not sexually dimorphic. So even the female is very brightly colored. So either the bright coloration is just ancestral and doesn't have anything to do with toxicity, or maybe we should be looking at the toxicity in these orioles.
Maybe we should in the orioles. Wouldn't that be cool?
So that's what we have about bright coloration and the evolution of toxicity in these birds.
Wallace would love you.
And so then the next question is even if toxicity isn't the ancestral character state, maybe there's some key innovation that encouraged or allowed the evolution of toxicity that we need to be thinking about. And the most interesting one to me is, how do Pitahuis actually tolerate such high levels of batrachotoxin in their muscle tissue? So we know that they're totally insensitive, and I'm going to just fly through this, and this is a lot of information to cover in the next 5 minutes. So the voltage-gated sodium channel, that's the protein that's targeted by batrachotoxin. This is an unusually large protein. It's a transmembrane protein that's about 260 kilodaltons. The mRNA is 200 amino acids, and its structure is very complicated, and there's between 24 and 27 exons spread out over sometimes hundreds of thousands of bases in the genome. This is what it looks like, and not only is it complicated on that level, but there's many different members of the sodium channel family, and they're expressed in different tissues and sometimes at different developmental stages. So for ease, and just to jump into this, we decided to focus on the skeletal muscle and the cardiac muscle, figuring that batrachotoxin probably doesn't pass through the blood-brain barrier, so whether or not these are insensitive might not be important. But the skeletal muscle and cardiac muscle are certainly exposed to the batrachotoxin in the birds, and these would be ones to look at for insensitivity. So if we look at this long, complicated protein, there's 4 different domains which are very similar to each other, but different. This is domain 1, domain 2, domain 3, and 4. The parts in green are actually the parts that are facing towards the center, towards the pore, which is probably where batrachotoxin binds, although nobody knows exactly where the batrachotoxin binding site is. And in past mutation studies have suggested that it's the S6, so there's these 6 different, within each domain there's these 6 different transmembrane portions, and it's 6 that faces the pore, and then there's also this thing called the P loop, or the pore loop, and that by mutating the S6 regions, you can sometimes achieve batrachotoxin insensitivity. So this is just a little bit about that binding site and how it works, but I've already mentioned that, so I won't mention it again. And so here, if you look at it, we think that batrachotoxin, and this is what the closed conformation of the sodium channel looks like. Nobody knows what the open conformation looks like. But when it's closed, it looks like this. We think that batrachotoxin is binding somewhere in the middle, and those green regions in the middle are highlighted here. So about a year and a half ago, a student from Butch Brody's lab contacted me because they're studying tetrodotoxin in newts and garter snakes. And she said, hey, I read this paper about pitahuis, and since we're doing the sequencing of sodium channels, we'd love to look and see how the batrachotoxin might bind and if there's anything interesting in the birds. So we got her all the samples we could get her, coached her through a few things that she would need to know about the birds. She did some amazing sequencing studies, but she was mostly targeting the S6 regions and a few of the exons that you can get out of the X6 regions. But she did find one site in the P loop of domain 4, and it's an isoleucine to methionine change that if you map it on the tree, it's sort of a basal corvoid group, and so it sort of encompasses all the birds that look like they have batrachotoxin and none of the birds that don't have batrachotoxin. And it's a significant amino acid change, and so we thought that this might be interesting. But there were some other things that were also interesting, and we've really covered such a small portion of the sodium channel gene that we wanted to hit it a little bit harder. So last summer, because of some work that I've been doing in Joe DeRisi's lab with viruses and some of the techniques I've learned, I said, well, let's just sequence the whole genome, and then we'll try and pull out these genes in silico. And they were skeptical that it would work, but I said, well, let's just try it. I'll front the money. And I did it myself in literally a week. And we extracted the DNA from variable pitahuis. We created Nextera libraries for long MySeq runs that were overlapping, and then also HiSeq runs that just gave us a ton of data. We trimmed them, did our quality control. Then we blasted these reads to zebrafinch sodium channel exons, and we pulled out the things that we thought were reliable hits. And then we used a program called PRICE, which is a DNA assembler that was designed to pull viruses out of huge genome sets. And basically, if you have a little bit that you think is a virus, it can find the reads around it and build out from it. And so we used the same approach to try and get the sodium channel genes. So using those things that blasted the zebrafinch, we used PRICE to assemble it, and then we went back to the longest contigs, and we tried to figure out if any of those actually had sodium channel genes, and it was beautiful. Usually we captured the entire coding regions with all the exons in between 3 and 5 contigs. And we had the upstream and downstream untranscribed regions as well. Then in December, you might remember that there was a paper that published 48 new bird genomes. And so now we had a lot of data to align with ours. And so in just the last couple months, we had put all of their data together with ours, and we have several of these looking at the different domains in the P loops and the S6 domains. And so this is just a snapshot of what the data is looking like right now. But basically, we're looking at any changes that are in these passerine lineages, but not anything else, and especially to make sure that things that have been tested for batrachotoxin sensibility, like chickens, that they are different from chickens that are sensitive to the toxins. And so, we've looked at several of these, and I won't go into too many details about it, but just last Friday, it turns out that we have a colleague at UCSF, who I've talked with for many years about doing something, but now with all this data, they're really excited to go forward. And he's very interested in trying to capture what that channel looks like in the open conformation. So with the batrachotoxin that we have on hand and some information from these birds about where to look for sensitivity, we're going to be trying to design some sodium channels that we can test in the laboratory. So, so where have we been in the last hour? So just to summarize, when you're out in the field and you find something as bizarre as a new poisonous bird, there's so many different questions to ask. And since it all started as a side project, you know, how deeply you can go into any one of these is difficult. But we've been able to learn a little bit about the chemistry of the toxins, what chemicals they are, how the toxins work, what the source of the toxin is, a little bit about how they evolved, and at least how the genus Pitahui actually looks, and suggesting some new avenues for research. We know a little bit about the adaptive function of batrachotoxin, but there's a lot more work to be done there. Something that we've totally not touched is the development of batrachotoxin. How does it develop in an individual from the egg onward, but also how does it get into the feathers? And then also this resistance to toxin, which we're starting to tackle now. And as all these talks go and all this kind of work goes, there are many, many, many people who we've collaborated, because this is way too much stuff for any one person to know about. So these are some of the collaborators that we've worked with and some of the funding that we've gotten. So thank you very much. And I think we might have a minute or two for questions. Yeah?
Have you found it in eggs?
We haven't. We've tried so hard to find a nest that was active, and we've
Been lucky enough to catch it. We've always tried to plan our field studies with, you know, birds might be breeding, but it's really hard to bird it down.
I guess that's the reason I wonder is that the nest predation is so high that that could be an interesting.
I think that's where the toxin is going to be the most valuable to the birds evolutionarily. And that's also where you can't get away from your ectoparasites if you're a little baby bird in the nest. That's where if you have the toxins on the outside, it would be super helpful. There was a group from Australia who had found a nest, and they watched it for quite a long time and published a nice little note about it. And they suggested that at least 3 and probably as many as 5 individuals were all caring for young, so that they're probably cooperative breeders. And that's something that we often find with poisonous things, is that they're often gregarious or cluster in groups. And if I remember correctly, they reported that the downy young were brightly colored like the adults, which would be unusual for a nestling. So, but we don't know about their toxicity. I can tell you that just from handling the birds and then letting them go, that if you taste your fingers or rub your eyes, you'll get that toxic effect. So I would imagine a bird with a brood patch sitting on the eggs, even if the toxin isn't already in the egg, that they would probably pick up enough of it on the outside. But that's still to be tested. Mm-hmm. We've never done any experiments, except for that one hooded pitahui that we injected with batrachotoxin. We've never done any experiments in the field to test for toxicity. And it's a little bit tougher thing to do these days to get IACUC approval to go injecting birds with known toxin.
Can you look at sodium channels? Can you send your sodium channel work to look and see if sodium channels in birds that aren't themselves carrying the toxin and be resistant to it?
It's definitely possible. It's going to take a lot of money to create a sodium channel that's a pitahui one in a bacteria that you can do expression studies is not going to be a trivial undertaking. So we would definitely want to start with something that we are pretty sure is insensitive first, but it's definitely something that's possible. Mm-hmm.
So I actually have 2 questions that are related. The first is, given that all of it seems to be that birds are picking up the toxin environmentally, just like most of the herbs are, in what sense are you looking at the evolution of something which is not genetically controlled, that is not sort of genetically part of maybe it's a propensity or something, but just sort of your thinking on that a little bit more, because it's something that in our lab we've been thinking about a lot with newts in terms of co-evolution stories when you're talking about co-evolution of something that's not genetically controlled in one of the players. And the other is, do you have any sense at all about consistency of toxin levels within individuals over time? You talked about tremendous variation, but it's all single point sampling. Do you have any sense at all whether, a bird that's toxic today is toxic tomorrow?
Yeah. So your second question first. The place where we do most work is Very Rada National Park, and it's been our old standby
from the capital of Port Moresby. And we visited there many different times of year, and every time we've been there, the pitahuis are poisonous enough that a single feather, and when you taste them, is toxic. So it seems, at least for those birds, that it's something that they do have year-round, and that they're relatively toxic. In terms of how much they vary throughout the year, that's going to take a bigger sample size and a much more complex study than we can do. And you probably know that most of the chemists that do these kinds of things, like, they have incredibly small sample sizes, and they're not interested in the kinds of sample sizes that we're interested in. And so it's hard to find. So we've never been able to do that yet. But I think a place like Very Rada would be a good place to get that more careful data. To your first question, I think it's really astute that you point that out, because so many of the species that are the classical species that we study as being chemically defended do get their toxins from some environmental source, and it really is more of a co-evolutionary thing, because it takes multiple partners for these. In that way, toxicity is almost like an epiphenomenon, because it takes multiple things to happen. And I think it's a very smart way to think about it. And I don't think I've thought about it as much as you have in that way, but I think it bears some thinking.
From a comparative physiological point of view, this sort of issue has come up in many, many different sorts of circumstances, not just with respect to toxicity. And there's an almost philosophical issue that I've wondered about forever, and I have no answer for it. And that is, if these guys can do it, and it's such a fundamental issue, like, how do if you can figure out a way of blocking something that's going to poison your sodium channels, I mean, that's something that could be really useful for lots of organisms. Why don't the rest of the world do it also? I mean, why is it restricted only to these very few isolated, presumably homoplasic kinds of occurrences?
Mm-hmm.
Do you have any thought about that?
Lots of thoughts, but I'm not sure that I have an answer. But I think a lot of it goes back to Brad's question, is that it does take, it takes some environmental collaboration to make these things work. So you have to have a source of the toxin, first. I think one of the biggest challenges that animals have to adapting these kinds of defenses is that you have to be insensitive to the toxin yourself. And those sodium channel genes are highly conserved, and tweaking them has real costs, and that's been shown in other things that are insensitive. So there's usually some other mutations that have to take place to take care of the damage that's done with the insensitivity.
Can it be an epigenetic phenomenon?
It can be. And you know what I also find very interesting is if you read those classic cases, from poison dart frogs to heliconius butterflies. Heliconius butterflies is not a good example. But many of the other things that we think of as the classic cases of aposematism, of gregarious behavior and toxic things, and even the toxicity themselves and how it works, they're very poorly studied from the kinds of standards that we would like to see. And so, I think all of them require a lot more study, and I think it would behoove us to find a system that you could really study well and get much deeper into those questions. And pitahui isn't going to be that, because it's a large charismatic bird that requires a lot of IACUC approvals, and it's hard to get to New Guinea, and it's hard to bring birds out. So, so it's not going to ever be a model system in my lifetime. But I think it would really behoove us to ask some of those kinds of questions in some species that can really be studied well. And the newt's probably one of the best examples of that. And I think that the work that Butch Brody and you guys are doing is amazing. And I think it's that kind of thinking outside the box and challenging the mainstream thinking.
It also looks to me as though you can't study these things without having an organism and an environment. And that's really pretty wonderful.
Well, and I think of the conservation implications, and you think of like how you really do have to have a healthy environment to support, not only that species, but all the things that that allows that species to persist in its environment. Because without the beetles and without whatever the beetles are eating and the healthy environment that keeps all those things in balance, you wouldn't have the pitahuis or their toxicity.